All right, I'm going to be talking about a new experiment on polymer chemistry called Emulsion copolymerization of sustainably derived myrcene and dibutyl itaconate. This is work done while I was on sabbatical at the University of Minnesota with a grad student, Ethan Gormung, and Professor Jane Wissinger's lab. So, we're really trying to think about polymers. We're basically trying to understand that polymers are critical components in our everyday lives. And I'll talk about a little bit about this NSF, Center for Sustainable Polymers, and the work that we're doing and they're doing over here at the University of Minnesota. But polymers are everywhere in our lives. Plastics are polymers, so you can see them in lots of medical devices, airplanes, bottle caps, toothbrushes, all sorts of places. Polymers are a part of our everyday lives. The problem is that they literally are everywhere, whether it's in our oceans, on the beaches, even in fish. So trying to think about ways to mitigate this plastic and these polymers has become really important. So here's the current paradigm, essentially, of how plastics and polymers come to be. Most of the polymers are created by monomers, but those monomers, those chemical starting materials, are from fossil fuel origins, which of course are not a sustainable source. We have a finite amount of fossil fuels left. They are then converted into useful materials. And then the other really harder issue is this end of life question. So really kind of booking these two ends. Where do we get our chemical feedstocks, our chemical starting materials? And then once we make a plastic or a polymer, what's its end of life like? How do we actually get rid of these plastics when we want to? So the University of Minnesota and the Center for Sustainable Polymers, they're working on answering those two questions. So we're trying to invent tomorrow's advanced polymers through integration of modern chemical sciences. So how can we use chemistry to answer those questions about where we get our starting materials for these plastics and then the end of life? So one of the major places is try to find biomass or plants or sustainable sourced starting materials. Could they come from corn? Could they come from biomass? Those type of things. Using chemistry to make useful materials, but then also once we're done using those materials, can they be broken back down into their chemical components or not be harmful to the native environment? So in this experiment, we're going to have a couple learning objectives. So we're going to do copolymerization, these learning objectives. We're actually going to do two reaction media. So it just means two types of reaction conditions. One is an aqueous emulsion. It's more like a micelle emulsion in aqueous environment. The other type is a solvent-free. We call that the bulk or a neat type reaction, but a lot of times it's referred to as bulk. Because we have two different types of reaction media, or reaction conditions, we're going to get two different types of materials or polymers. In the aqueous emulsion, we're going to get cross-linked materials, and we'll talk more about those. In the bulk, we're going to get linear polymers, which have a visible viscosity change. Because again, we're different, we're going to be able to do a pH isolation of this polymer by a pH change, a very visible phenomenon, and we won't be able to do NMR spectroscopy, but with our linear polymer, we actually will be able to do NMR spectroscopy and try to use NMR to understand how big of a polymer we have, the molar mass. And then finally, because this is cross-linked, it actually forms a gel. And we're going to look at that gel and see how it absorbs different organic solvents and organic compounds. And finally, with the solvent-free, we'll also be able to figure out how much monomer of each type of monomer was incorporated into the polymer. So taking a little closer look at the chemistry. So our two starting materials in both situations. First is both myrcene, beta-myrcene, this compound here, which is derived from beta-pinene, which is a naturally sourced compound from plants. And undergoes a couple steps in pyrolysis, and we get to beta-myrcene. Glucose is actually the starting backbone for dibutyl itaconate, which glucose undergoing fermentation actually gets us to this starting material. So these are sustainably sourced starting materials. So when we do an emulsion, we're going to create like a micelle, so we use a base and a long chain fatty acid type with a polar head and a non-polar tail. And those are our emulsifiers, helping create that micelle. And this is our initiator, our radical initiator. So we're going to do this in water at 70 degrees, so radical initiation. We're going to react with these alkene subunits and do a radical polymerization. So initially it's going to happen, we have our initiator start out, and we're going to have a mixture of myrcene and dibutyl itaconate. So myrcene and dibutyl itaconate basically 50, should be about a 50-50 ratio if we had even amounts of these, they react at the same rate. Interesting though, when you're doing emulsion, right, these emulsions, you actually have cross-linking sites, so places where this could actually become cross-linked, and we'll talk a little bit more about that in a second. The other type, the bulk type, again uses myrcene and dibutylconate. No emulsion this time. We're going to use neat, so solventless. So just take these two liquids. We're going to put benzoyl peroxide, again another radical initiator. And it's going to be neat at 70 degrees for 1 week. And again, we're going to get, instead of cross-linking, we're going to get a linear polymer, straight chain polymer. And that's going to incorporate myrcene and dibutylconate in equal ratios. If this was a 1-to-1, these would be an equal ratio there. So when we talk about cross-linking, so this is that emulsion polymerization. What does this look like kind of in a figure? Right? So we have a number of monomers, right, the red and the blue, and they're in a random statistical arrangement, along chains. And what happens at these cross-linking sites, these alkenes, those alkenes can react with other alkenes in a whole different long chain, and these create cross-links. And this makes the material much more insoluble and rigid, and it also allows it to swell. So we won't be able to dissolve this material, this crossing material, in anything, but we will be able to use it to look at swelling. So again, at the cross-linking sites on different chains, these would start pulling these chains together. So in week 1, we're going to do an emulsion copolymerization. So you're going to make a cross-linked material, and you're going to precipitate that material in ethanol, and then we're going to dry it over the vacuum oven over the week. So that's the first step. You're going to do the emulsion copolymerization, that type of reaction condition. The next step, you're going to do a bulk polymerization, which is just solvent-free on a little bit of smaller scale, and we're going to use that for NMR analysis for the following week. So that's the first week. You're going to do those things. This takes about an hour. This you just set up and leave it. In week 2, the first thing you're going to do is do an NMR of your neat copolymer. So we're going to do that first. The second thing, you're going to use our cross-linked polymer from the emulsion chemistry and do some swelling studies with dopants. So that polymer can actually possibly could select for different small molecules. And you can imagine about drug delivery systems or other ways in the environment, if you wanted to pull certain organic molecules out of the environment, you could use a cross-linked material to do that. So we're going to do that with a stock solution spogged with toluene and benzyl alcohol for 45 minutes at 40 degrees. And you're also going to, after you're done, you're going to take an NMR analysis of the solvent that's left over and see what is in that solvent. And that'll be explained a little bit more in a second. And finally, you're going to set up a qualitative solvent comparison where you're going to take some more of that cross-linked polymer and take a look at it in chloroform, THF, acetone, DMSO, and water. So you can see all 5 of these solvents have different polarities, perhaps, and see how the crossing material likes certain polar or non-polar solvents. So to kind of go back to that bulk polymerization, we're going to take a look at an NMR here, and the most important thing to look at in this case is we're going to look at these alkene protons. That's really what we want you to focus on. They're all labeled here, right? And ideally what's going to happen is these, especially these up here, they're not going to be present after the bulk polymerization because they're going to have reacted, right? So you're going to look for the disappearance of some of those peaks. Other starting materials, dibutylconate. Again, we're looking for these alkene protons right here. And in this case, we're also going to look for protons 10 and protons 14, which are right here. We're going to be looking for these to make sure to see after the polymerization, is there any starting material left? All right, so what is the final NMR of the polymer going to look like? So that's shown here. So here's the NMR of the polymer. And you can see left over, there's a little bit of leftover starting material dibutylconate. You can see both there and here. And if you see there's no observed myrcene left. You also notice in the polymer, 4A here, there's 2 peaks, is very broad because it's a big, large molecule. And also look for B, the 2 H's adjacent to the ester, those are located here. And notice how big these are, obviously because there's a polymerization that's taking place. The NMR, the assignment we have for you, will walk you through how to do all these calculations using N-group analysis, using some of this initiator peaks. It's all explained later. Just want to make sure you've seen and know where to look for things. All right, so now going on to the swelling standard. In that case, we took a bit of our cross-linked polymer. So this is the stuff that we crashed out that first day. And we're going to see we have two molecules, benzyl alcohol and toluene. And you can see these are similar molecules. Both have 6 carbons, same number of hydrogens, except now benzyl alcohol has an alcohol group. So we would guess this is a much more polar molecule, and this is a non-polar molecule. And what you're going to do is you have an NMR of a standard solution that's been made for you, and the ratio of these hydrogens should be 2 to 3. It's a 50-50 mixture. It should be 2 to 3, that ratio. What we're going to test to see is, does the polymer that you've created, the cross-linked polymer you've created, select or absorb one of these more than the other. So that setup's going to look like this. We're going to have our stock solution's been made for you, where it should be a 2 to 3 ratio. So we'll take an NMR of that pre-stock material, and you'll be given that NMR. You're going to add in your cross-linked material. That cross-linked material that cross-linked polymer is going to sit there for 40 minutes at 45 minutes at 40 degrees. And what's going to happen, it's going to swell up. And what we want to see is, does the polymer, or does one of these molecules, is it more attracted to the polymer than the other? And you'll know whether or not it's attracted more than the other, because you'll take whatever solvent's left over, you'll take that out, and you'll put it in an NMR tube. And you'll compare it to this NMR and see in the resulting solution, did the ratio of these change? And if it did, if there's all of a sudden there's less of this, you'll know by NMR to see that. And that'll tell you something about the nature of this polymer and what kind of molecules it's attracted to, polar or non-polar. Again, just to emphasize, you should have a ratio to start off 2 to 3. And if this ratio changes, that will tell you something about whether one of these molecules has now been absorbed into the polymer. Finally, part E, we're looking at the set up the qualitative solvent comparison, looking at 5 different solvents of different polarities. You'll simply have those little bits of your polymer in little vials, and you'll have different solvents placed in all of them, and you just qualitatively look at it and see, is my polymer swelling or is it not? And then it'll tell you something about the nature of the polymer itself, whether it's attracted to polar or non-polar organic compounds. I hope you enjoy this. We had a lot of fun working on it, and please let us know if you have any questions.